discovery that DNA is the genetic material, the race to uncover the three-dimensional structure of DNA began. The arrangement of the bonds in a DNA polymer was already known. Each monomer unit, or nucleotide, of the polynucleotide chain is made up of a phosphate group, the sugar deoxyribose, and a nitrogenous base. The phosphate groups and deoxyribose sugars form the backbone of the chain. There are 4 nitrogenous bases: guanine, cytosine, adenine, and thymine, abbreviated G, C, A, and T. The Austrian biochemist Erwin Chargaff found that the amounts of adenine and thymine found in DNA were always about equal to each other. Determining the three-dimensional structure of a DNA molecule required data from several groups. In the early 1950s, while working in London, Rosalind Franklin produced photographs of the X-ray diffraction patterns of DNA fibers. In 1953, from Franklin's X-ray photographs, an American James Watson and an Englishman Francis Crick determined that DNA is a spiraling coil called a helix. They were also able to tell that the helix was about 2 nanometers wide. This fit with DNA being a helix of 2 polynucleotide chains, commonly called a DNA double helix. In a DNA double helix, each width of the double helix also helps scientists understand Chargaff's rules. Adenine and guanine have 2 rings. They are called purines. Thymine and cytosine have only 1 ring and are called pyrimidines. To fit the width of the double helix, a purine on one strand must pair with a pyrimidine on the other strand. Chargaff's rules state that there is the same amount of adenine as thymine, so adenine pairs with thymine. Similarly, guanine pairs with cytosine. Another interesting aspect of DNA structure is the orientation of the 2 strands in the double helix. The strands run in opposite directions. This is called an anti-parallel orientation. The carbon atoms in the deoxyribose sugar are carbon 1 to carbon 5. A phosphate group is bonded to the number 5 carbon, known as the 5' carbon, and another phosphate group is bonded to the number 3 carbon, the 3' carbon. For simplicity, we'll place one strand with the 5' carbons all oriented to the top of the screen and the 3' carbons to the bottom of the screen. Since the other strand is anti-parallel, all of the 5' carbons will face the bottom of the screen and the 3' carbons will face the top of the screen.